for small animals. So for small organisms, for protozoans, for little tiny animals like rotifers and things like that, viscosity is extremely high compared to the counteracting inertia. And so the other forces that act on things are inertia, which is a general tendency to keep moving if you're moving, and turbulence. So in a vacuum, if you launch a spaceship out in a vacuum, even if it doesn't have any propulsion, once you stop the propulsion, it'll just keep going at that same speed forever and ever until it collides with something. Because there's no resistance, there's nothing to slow it. That's in basic inertia. Of course, on Earth, we have in an atmosphere or in water, you have viscosity and turbulence that tends to that friction that tends to counteract inertia. And inertia is higher the more mass you have, for example. The other factor is turbulence. Large-scale turbulence happens when the fluid does not flow smoothly around you, and the smooth flow breaks up and creates low-pressure areas in the direction away from which you're moving. So as you move through the fluid, behind you can have turbulence. That turbulence creates a low-pressure area that basically sucks you back. It counteracts your tendency to move forward. If you're very small, turbulence is pretty insignificant. It doesn't really matter very much. And if you're very small, you don't have much inertia either, because that's dependent on your mass. So for small animals, things like protozoans and small aquatic organisms, pretty much the main force is viscosity. So for something like a paramecium or paranema or something swimming through water, to them it's basically like swimming through molasses. It's all about viscosity, and they have to work hard just to overcome that viscosity. But they don't generally encounter much turbulence, so they don't really have to worry too much about being streamlined, which is one way to reduce turbulence. But they do have a lot of that friction. They do have to overcome that viscosity. And because they're so small, they have essentially no inertia. So if they stop working, they stop instantly. So you can start instantly, you can start up instantly, you can stop instantly. You don't glide, because the gliding is basically the result of inertia. So if you see a protozoan moving across your microscope slide, you know it's working its little butts off, or its little cilia off, to be able to keep doing that swimming. If it were to stop moving its cilia, it would come to a dead stop instantly. Big animals, on the other hand, have kind of the opposite effect. In large animals, you have proportionally less surface area, so viscosity tends to not be as important relative to the effect of turbulence. You're moving much larger volumes of water around you, and so that creates more turbulence behind you. And if you're big, you also have more inertia as well, the tendency to keep moving. And so large animals tend to have to worry much more about drag turbulence. And so for large animals, it actually makes sense to be streamlined if you're going to be moving reasonably quickly. On the other hand, that also means that for large animals, it takes a lot. They have to overcome inertia. If you're not moving, it takes effort just to overcome the inertia and accelerate. And then if you are moving, that inertia can allow you to continue moving for a while, even if you stop applying force. You can basically drift or float. And so for larger animals, they actually need to actively, if they want to come to a quick stop, they may actually need to spend energy on undoing that inertia. So basically, your size is, and we're not going to get into a lot of the physics involved here, but what I do want to kind of impress upon you is that the size of the organism, and kind of really impress on you the importance of body size. It has really dramatic effects on lots of aspects of an organism's biology. But size is going to affect how you see the world. If you're really tiny, you're going to be swimming through, it's going to feel like you're swimming through molasses. You have this thick, high friction environment that as soon as you take a break, you're going to come to an instant stop. If you're a large animal, you take a rest, you just kind of keep gliding for a while, and you'll slow down because viscosity and friction does have its effect, but it'll take a while because you have inertia. So very different sort of physics, and therefore very different ways organisms need to respond to that physics. All right, anyway, with that little bit of background out of the way, let's look at how organisms actually move. There are essentially, what do we have? 6 different types of locomotion here, found in both animals and protozoans. You've seen a couple of these already in labs. So we have amoeboid motion, where you basically extend out pseudopods and you crawl across the surface relatively slowly. You have the motion that is produced by either cilia or flagella. Since these are very similar structures, the basic way they produce motion is relatively similar as well. There are some differences, but the basic principle is the same. You're basically using a cellular-sized appendage to push against the substrate, against the water. You can have hydrostatic propulsion, which is basically where the muscles act against a hydrostatic skeleton to flex the body. So you're swimming, or you're crawling, or you're pushing your way through a substrate by expanding and contracting or flexing the body. And that flexing is done because the muscles are acting against a hydrostatic skeleton. That is a fluid-filled cavity where the internal fluid pressure provides a structure that's semi-rigid that muscles can act against. You can also move by either using limbs or even just the body as a whole against a more rigid skeleton, particularly an articulating skeleton, where the muscles act against rigid components of an appendage. And so that's basically what us vertebrates do. We have limbs, we have a skeleton that articulates, that has joints where it moves, other places where it's relatively rigid. That's the way arthropods move as well, except their skeletons on the outside. It's the same kind of principle. So you're using limb movement. Those limbs can be paddles to swim with. They can be legs to crawl over the substrate with, and so on. And then finally, you have jet propulsion, where you forcefully expel water or some other, I suppose maybe if you have really powerful farts, you can expel air gases and move through the air. But jet propulsion typically expelling water, and where you move in the opposite direction from which you're expelling water. So that's used by some mollusks like squid and octopi and some kind of scallops, for example. And probably a few other things here and there. All right. So let's do amoeboid motion first. So amoeboid motion is carried out by amoebas. Obviously, that's where the name comes from. It's used by a number of other protozoans, as it seems to have evolved in different protozoan groups. And it's also used by the majority of animals in specific cells within animals. So all of you have cells that are capable of amoeboid motion and use it. You have your white blood cells, the white corpuscles that are traveling through your body, through your bloodstream and through extracellular fluids, seeking out harmful substances, bacteria, and so on. And they largely move through using this amoeboid motion as well. We find that even the most primitive animal sponges have amoeboid cells that move through their body like amoeba do. So the majority, if not all animals, have cells that carry out amoeboid motion. So what is amoeboid motion? Well, it basically involves a formation of a pseudopod, a temporary extension of the cell that extends outwards, and then the cytoplasm flows into that pseudopod, and the cell creeps in that direction. We have some idea of how it works, but it's not 100% understood exactly how the mechanism works. So here's a video. This just shows an amoeba here, and you can see the pseudopod extending outwards. You can see the cytoplasm flowing into that pseudopod. Move this slide down here. And again, as it extends outwards, you can form new pseudopods here. And the rest of it, and as the cytoplasm flows, the back end is basically being drawn forwards here. You're extending some pseudopods in a couple of different directions. Okay. So it's a pretty slow movement, but it gives you nice stability, for example. And you can use those pseudopods. Probably the main advantage here is that you're using those pseudopods for other things. Those pseudopods can engulf large food items, for example. And so then if you're producing pseudopods anyway, you can also use that to move around. All right, so how does this work? Well, this is what we think happens. That is in the, so here's a pseudoplasm, here's a piece of an amoeba, a section of the amoeba, and the cytoplasm is kind of subdivided into ectoplasm near the plasma membrane and endoplasm further in the interior. The ectoplasm is generally loaded up with actin fibers that help kind of support that shape. It's a temporary arrangement, so it's not a rigid shape. And what happens is if the amoeba wants to move in this direction, in the direction that it wants to go, it basically uses a little enzyme to break down those actins fibers into tiny little pieces. And as you break those big fibers into little pieces, you have more solutes. You increase the number of solutes, so you increase the osmotic concentration where you're producing all of the little actin pieces. And that increased osmotic concentration basically sucks in water because you have a high concentration of solute. Water follows, gets drawn in, and that flow of water then pushes in that direction and causes the pseudopod to extend up. That buildup of fluid that's produced by those broken up actins pushes out the pseudopod, and then to stabilize that pseudopod, you repolymerize the actin, you join it back together, and you keep it somewhat stable. You can then extend it further by breaking this down even more, and so on. So this is all happening very rapidly as a series of chemical reactions. At least that's the sort of basic method in which we think this whole process takes place. All right. Any questions so far? All right, let's go on to cilia and flagella. Pretty much all animals have cells that contain either cilia or flagella. The vast majority of animals have mobile sperm. Now, there may be some exceptions, but most have mobile sperm. That means half of all animals, roughly speaking, at least half have flagella. And cilia are also fairly common in most animals as well. You have cells that have, some of you have cells with flagella, but all of you have cells with cilia, and you have them lining your respiratory tracts, for example. Many animals are covered externally. Many small species, things like flatworms and gastrotrix and rotifers, have external cilia covering their bodies that they use for swimming. Many larger animals have ciliated larvae. So those early simple larvae locomote because they mainly have to be, a handful of cells, but those cells have cilia, which allows them to locomote. So again, that's why your larvae is very different. While an adult might swim with paddles or something like that, the larvae use cilia. And so cilia and flagella are both pretty much almost universal in the animal kingdom. They're also found in many kinds of protozoans and other organisms as well. We're grouping them together because they're basically built the same way. They have the same internal structure, which is diagrammed here. So this little diagram shows a close-up of, could be either a cilium or a flagellum. And so if you slice it in open, what you would find inside is 9 pairs of microtubules. So each little, what looks like a little bug here or something, is the round circles are the microtubules. There are 2 that are they're in pairs, they're bonded together, and then extending out from one of those microtubules are these projecting arms called dynine arms. And the dynine arms from one microtubule basically attach to the one pair of microtubules attached to the adjacent pair. And those dynine arms can connect and disconnect, and they can move, so they can essentially basically cause the tubules to slide past each other. They attach, move, swing, unattach, grab, move, and so the 2 tubules can crawl past the 2 different pairs of tubules can crawl past each other, and as they do so, one side of the cilium or flagellum can shorten and bend compared to the other side. So it's that movement, this relative sliding of those microtubules, that causes a filament and causes a flagellum or cilium to bend, and that's the basic way that cilia and flagella move. And we're not going to worry about the basic, the sort of the stuff at the within the cell here, but it gets a little more complicated. All right. So what is the difference between cilia and flagella? Well, typically cilia generally are shorter. They are less than one. So by definition, they are less than one wavelength long. So that, and the wavelength is the length of one sweeping range of movement in the arm. For cilia, they're less than one complete wavelength. And typically, now there are exceptions to this, especially among animals, but among protozoans in particular, cilia are almost always in patches, in relatively large numbers, with bunches of cilia together. Not necessarily uniformly covering the protozoan, they might be arranged in rings and other kinds of patterns, but there are always bunches of cilia together. In some animals in which they are multicellular, there may be just a single cilia associated with each individual cell, but amongst protozoans, they're always in groups. And if they are in groups, the movements of those cilia are highly coordinated. Within the cell itself, there are mechanisms so that adjacent cilia are essentially moving in exactly the same way, and there may be that movement may be timed so that it basically can sweep across the surface of the protozoan. Compared to cilia, flagella tend to be longer. They're often more than one wavelength long. They don't always have to be, but they're usually more than one wavelength. They're usually only present one or two or three, although you guys saw an exception, right? What was that? Remember? The trichonymphas, yeah. So the parabasalids and the parasitic ones. Some of those have, like the ones the trichonympha have, in some cases, many flagella. What makes them flagella is they're not coordinated the same way cilia would be, and they're much longer. They're longer than the wavelength of movement. So it's also possible, in some cases, the flagella might have little projections that stick out to look like little hairs on the flagella itself. That increases surface area and basically increase effectiveness. Like instead of like rowing your boat with a stick, you're rowing your boat with a paddle. And the movement of waves in flagella can start either at the base or the tip, so the flagella can push or pull. I think I have slides of that coming up. All right. So in cilia, basically the cilia alternate with a power and recovery stroke. So if you look at it from the side, this is the top arrow here shows the direction of the organism's movement. It's moving from left, so the organism here is moving in this direction, and the cilia are bending this way. This is the power stroke this way. It's pushing against the water, and as it pushes against the water, the animal moves in the opposite direction, so it moves in that direction. But then you have to return the cilia to that position so that you have the recovery stroke, where the cilia folds close to the cell surface and basically moves this way. This picture at the top is actually a little bit better because in 3 dimensions it's very common for the cilia to kind of bend around so that you're close to the surface, you're not pushing against much water. And so here's the power stroke, the cilia, so you're moving to the left. So this is the power stroke here, pushing the cilia that way. Then you have the recovery stroke where it bends down, twists around until it's basically back in position for the next power stroke this way. And something like this protozoan here, the ciliated protozoan, those power strokes are basically synchronized. So all of the so you can see the kind of wave here. So you can see where the peak. Those are the ones in the peak power stroke. These are the ones in the recovery stroke here, and those power and recovery strokes kind of move in a synchronous pattern across those rows of cilia in a coordinated fashion, so the animal is always kind of swimming. In contrast with flagella, and it usually depends on the organism, so they'll usually use one or the other type. Flagella can basically start at the base, the movement can start at the base. So you basically start here, If you had the flagellum straight out, it would push this way, and then it would push in the opposite direction. As it pushes to the side, as the flagellum pushes here, that movement moves further back, and so it pushes against the water this way, and it pushes against the water that way. While it's over here, it's pushing against the water this way and that way. Because you have a complete a wavelength here, you're pushing in both directions equally, so the lateral movements cancel each other out, and all that's left is the vector pushing the water in the opposite direction, which means the animal or the protozoan or sperm is moving in this direction. And remember, you don't have to necessarily be moving either. You can use cilia and flagella to move fluids past you. From the organism perspective, it may not really make that much of a difference. You're just moving fluids. Whether if you're not attached to anything, you're going to move through the fluid in the opposite direction that you're pushing or pulling. But if you're attached to a substrate, you will be moving water through you. And many animals make use of that. The sponges we'll see in lab today make use of that. All right, and so you can also have the movement start at the tip, where the wave moves down this way. So in that case, the flagellum is essentially pulling the animal. So depending on where the undulation starts, you can either push, like we think of like with the sperm cell, or you can pull the animal, as you saw with the Paranema last week in lab. All right. So, I think I've already said many small animals, including things like rotifers and flatworms, and many larvae of other animals rely on cilia for locomotion. But in addition to cilia, most animals use muscles. So muscles are not found in sponges, although sponges do have some sort of contractile cells, but they're not true muscles. We'll get into that when we get into sponges. But almost all other animals, including the cnidarians, anything with tissue-level organization, among those tissues include muscle tissue. And muscle tissue can contract, but if the muscle tissue is not attached to anything solid, it just shrinks up the animal, it just causes it to shrivel. So in order for muscles to kind of be effective, to create effective movement, and we're out of time, aren't we? Yes, we are. Let's actually stop. Forget I said anything about muscles. We'll stop with cilia and flagella. I should have probably taken a look at the class before. We'll worry about muscles, and then in particular that will get us into skeletons, which we'll talk about